Well, we were talking about the fluid mosaic model of the cell membrane, and we were identifying the types of molecule which assemble together to constitute the complete cell membrane, and then cell membrane, which is part of a big ship. So phospholipids are here. Hydrophobic ends are inside and hydrophilic ends are outside. They are facing water. Cytosol inside and extracellular fluid outside. And next molecules are so this constitutes the whole assembly known as phospholipid bilayer. So this name, the fluid mosaic model, it comes from the thing that it's kind of the whole aqueous material or the fluid that that has embedded or micelles in it, and all the flow of the materials from inside to the outside of the cell and outside to the inside, that is all controlled or regulated through the pores or the molecular structure of the cell membrane. These are the cholesterol molecules. Cholesterol is hydrophobic. That's why it is embedded inside the hydrophobic, in between the hydrophobic layers or hydrophobic chains, carbon chains of the lipids. Cholesterol, this is not facing water outside or inside. It's embedded inside the hydrophobic ends of the lipids, fatty acid tails. And then there are types of blue moieties. These are all proteins, and there are different types of proteins. In protein section, we will talk about how there are different proteins and what are their different functions. These are recognition proteins. Recognition proteins mean they recognize particular type of protein, and only that protein can embed or can attach with those recognition proteins. This is kind of a key lock theory. Okay, so in this, only the particular protein that is recognized by this recognition protein, it can bind with this protein. In these days, you are also learning about coronavirus. Coronavirus is also kind of a protein, and the structure of that protein is kind of a crown on the ball. There are multiple these type moieties, this type of structures, and वो जो कोरोना का बॉल है वो आके इन रिकग्निशन प्रोटीन्स पे अटैच हो जाता है एक पॉइंट से, और इट गोज इनसाइड. And how it goes inside, that is by another type of protein. We will see it. Receptor protein. Receptor protein are the again key lock kind of concept, that receptor protein, it receives the incoming protein at this site only. This is called as binding site of the receptor protein. And there are transport protein. Through these transport proteins, there is channel in between, and through these transport proteins, the outside protein, such as this protein, it can be moved into the cell, into the cytosol. There is a pore there. These are kind of glycoproteins, which the proteins which are, this is protein structure, and there is let's suppose this is a protein structure and this is a glucose molecule. These are combination with the carbohydrates, so these are glycoproteins, different proteins. So vice versa, protein, carbohydrates, carbohydrate chain is there. These are protein filaments all scattered inside the cytosol. So all this constitutes the cell membrane components. Again, in detail, you can see we will talk about the lipids, actually, the phospholipid or lipid bilayer. Bilayer comes from this first layer of lipids and the second layer of lipids. Hydrophilic ends here, hydrophobic ends tails here, and the extracellular fluid watery environment is in touch with this one, with the hydrophilic end, that is actually the glycerol part of the fatty acids or lipids, and these are all phospholipids. Coming here, hydrophobic tails. Hydrophobic tail is the pink part, and hydrophilic heads are coming from this part. You can see it clearly. Okay, so that is how the lipid bilayer structure is constituted and is the active part of the cell. It controls the cellular activities that are constituting the whole fluid mosaic model. And this is why, due to the hydrophobic structure, cholesterol or any of the non-polar component will be trapped here inside these states. Why do fatty acid tails of phospholipids point towards each other in the membrane? Let's have the options. They are non-polar. ये non-polar होती हैं phospholipid tails, so they are hydrophilic. No, non-polar, it aligns with hydrophobic in nature. Let's see the second example. They are non-polar, so they are hydrophobic. Seems fine. Number third option is they are polar, so they are hydrophilic. No, phospholipids tails are not polar and they are not hydrophilic, so this is also not correct. Number four, they are polar, so they are hydrophobic. Again, polarity is linked with hydrophilicity, not hydrophobicity. So option 2 seems to be correct one. Let's see. Yes. The next is membrane components or proteins. There are other types of components in the cell. These cell membrane, which are different types of proteins. These are constituted with amino acids, and amino acids are linked together to form peptides. And the final shape of the protein determines its function. If it is having pore inside, so it is kind of a transport protein. What conditions can denature proteins? Change in the pH. If you heat any of the protein, it also denatures. I suppose the egg protein is albumin, egg white, and it's colorless when it is uncooked. When you cook it, it's white. So there are multiple things which can denature the proteins. So you need to make some predictions in this slide. What are some characteristics of molecules that can diffuse directly through the lipid layer of the cell membrane? This is also your assignment as well. What are some characteristics of molecules that will need the assistance of carrier proteins to move through the membranes? You better solve this after the listening this whole presentation, and then you will answer these questions. You can take help of Google or whatever, but before that you try to do it yourself. Transport of materials through the cell membrane. It happens through passive transports. It's without the use of energy. The examples are simple diffusion, facilitated diffusion, and osmosis. Energy consuming transport or active transport, these are through exoendocytosis as well. Diffusion, simple process. When you put a drop of dye in water, it spreads itself and the whole mixture is now at the same color. Similarly, let's take the example of sugar. When you put few crystals of sugar in water, after some time through the process of diffusion, it gets dissolved, right? So this is another example of diffusion. You are familiar with this. So diffusion across a membrane, lipid soluble molecules and oxygen or carbon dioxide